Hi everyone, so in this video I'm going to go over glycolysis and pyruvate processing. And so just before I start, I just want to give a general overview of what is happening in cellular respiration. So we know that cellular respiration consists of glycolysis all the way until we get to oxidative phosphorylation, right? And so really the main idea is that glycolysis is starting with a reduced glucose molecule, and then eventually what we want to do is we want to get all of the electrons from this highly reduced molecule to be able to put this into oxidative phosphorylation and then get our ATP out. And so ultimately the goal is, okay, so the cell needs to set up a way to extract all of these electrons from this highly reduced molecule and be able to store those electrons in a way that they can be moved around easily. And then we need a place where we can basically cache in these electrons and get our energy output. So in this video, I'm just going to talk about glycolysis and pyruvate processing, but then in my other videos, I'll talk about the other steps in cellular respiration. So with glycolysis being the first step, so glycolysis is a process that occurs in the cytosol of the cell. And glycolysis is really the process of going from a glucose molecule to two pyruvate molecules. And so we can sort of break it down into phases. You don't really need to know the details of this, but I'm just going to go through the phases just for completion. So in phase 1, so this is when we're prepping our glucose molecule to extract electrons from it. So we actually put in 2 ATP at this stage. And this is through substrate level phosphorylation. So I might go through this in another video, but if I don't, please know that you have to know what substrate level phosphorylation is and that it is very different from oxidative phosphorylation. They're completely different processes and they happen in very distinctly different ways. So the main idea is that substrate level phosphorylation is happening using an enzyme, where oxidative phosphorylation, as we saw, is using the electron transport chain, a proton gradient, and ATP synthase. So it's a whole complicated, very orchestrated process, whereas substrate level phosphorylation is really just using an enzyme in order to phosphorylate something. So in phase 1, we're inputting these 2 ATPs, and that phosphorylation is occurring by substrate level phosphorylation. And then in phase 2, we're not inputting or outputting anything. This is where we actually split the glucose molecule in half, so that in phase 3, we basically now get our energy output from each of those halves. So for I'll say for 1/2 of the glucose, we're actually going to get 2 ATP, and we're going to get 1 NADH. And so NADH here is really what has now electrons on it. So ATP is our main form of energy, but NADH also has energy in it in the form of electrons. It's very reduced. So that's something that'll pay off later when we talk about oxidative phosphorylation. So if we want to talk about the overall inputs and outputs, because I'm guessing that you'll likely have to do these for the exam. So if I draw our glycolysis, so our input obviously is glucose, right? Glycolysis itself is the lysis of glucose, so the splitting, the cutting of glucose. And our output is going to be pyruvate. And then just a reminder that when you're drawing arrows for inputs and outputs with energy molecules, you always want to draw them as dotted lines. So we're going to be inputting NAD plus and outputting NADH, right? so the input NADH that we're reducing with electrons in phase 3, and we're getting NADH out of that. Sorry, I don't know what I said, but I meant NAD plus is being reduced by putting electrons onto it and then becoming NADH. And then we're also going to add ADP as an input. So this is a net input because notice how we actually have to put in 2 ATPs, but then in total we actually get 4 ATP out, right? Because we get 2 ATP per half of a glucose molecule. So overall we put 2 in, but we get 4 out. So the net gain is 2. So overall we're putting ADP in and we're getting ATP out. Okay, so that's our glycolysis input and output diagram. And so now I'm going to go through pyruvate processing. So pyruvate is the output of glycolysis. And so it actually occurs now inside the mitochondria. So there's actually a transporter that carries pyruvate across the mitochondrial membrane, and so now we are inside the mitochondria, and now the job is basically to prep pyruvate. So the main idea. So now we're in the mitochondrial matrix. So the main idea is really just prepping pyruvate for the TCA cycle. And so you don't really need to know a lot of these details, but basically we're going from pyruvate to acetyl-CoA, which is our input, one of our inputs for the TCA cycle. And through this process, we are also reducing an NAD plus to get an NADH. We're releasing a carbon dioxide molecule, which this is a hint, right? If I'm releasing something oxidized, that means something else had to be reduced. So just a hint that generally when something is being oxidized, something else is being reduced. And in this case, we're reducing that NAD plus, right? We're donating those electrons to make more NADH. And we're also going to input coenzyme A, which is just this this little thing that's like attached to our acetyl molecule. So so that's basically the overall. I can draw the input and output, but it's basically what I just drew. So pyruvate, oh my gosh, pyruvate, it goes in, you get acetyl-CoA out. You also have to input a CoA molecule. You also get CO2 as an output. And then for our high energy molecules, we're inputting 1 NAD plus, and we're reducing that to get an NADH out. So overall, we're releasing something that's a really oxidized form of carbon, which is CO2, and we're also reducing NAD plus to get more NADH, which we will later cash in when we get to oxidative phosphorylation. So hopefully that gave a good general idea of what's happening in these first 2 steps. And in the next video, I'll talk about the TCA cycle and oxidative phosphorylation.